Hi, my name is Charles Warden, and I'll be discussing sharing and reporting genomic results and data. A lot of this content has been previously posted as blog posts, but some of it will be new. So I'm going to split this up into three main categories. The first is FDA MedWatch reports. So FDA MedWatch is a system for reporting adverse events, and in particular, I believe I'm considering these genomic results to be like a medical device. The second is PrecisionFDA. So this may be limited to a somewhat more limited number of individuals, but it relates to sharing large genomic data, which you might call the raw data. And the last section will actually relate to a few different things, but one thing that I'm learning about now that I think is very interesting is NIH ClinGen Genome Connect, which includes a way to be able to upload the PDFs of your genetic reports, and I'll describe more about that at the end of the slides. So for FDA MedWatch, this is what you probably see when you look for FDA MedWatch, and then the first step will be to click the red button that says report a problem. Now, my understanding is that part of the reason FDA MedWatch was created was to have a system so that you could report issues where you might have a recall or reversal of FDA approval. But what I think is really important and useful is that the products don't have to be FDA approved for you to report an adverse event. And so for these genomic results, I believe none of them are FDA approved for the genomic part. So that is something that I think is of use in general, so that if you want to have some way that, assuming the FDA staff approve your report and make a publicly identified report, it allows you to share those experiences. Other people can look for those experiences in a database that I will show at a later slide. So there are multiple ways you can submit FDA MedWatch reports, but I'm going to be talking about the consumer patient form. And it's a multi-part form, and I'll go through this in a little bit more detail, but essentially what I'm saying here is that if you submit a report and it's accepted, I believe you should receive a notification. If I'm remembering correctly, maybe it's on a matter of months or so. But if you don't receive a report, then I don't believe you get notification, and I don't believe I've ever received an explanation of why it was not accepted. And to be clear, the FDA does not want you to resubmit the same report again. So if it's been more than a few months and you haven't got notifications been accepted, then you can consider thinking about ways you could collect more evidence or explain yourself better and resubmit. But I would take some time and think about what could be changed in the report. And the next slide will relate to the FDA MAUDE database, which is if the report gets accepted, then that's where a data identified version will be posted publicly. But let me kind of go through these past few slides in real time. So if we go to Google and we look for FDA MedWatch, then this will get us to the slide that the page that I described. So we click report a problem, and you scroll down, say consumer patient, and each of these dots is a different step in the process. I believe you have to fill out the form in order to get to the next page. So I'm not going to do that, but you can kind of see, for example, here's a part of the form where we have about 4,000 characters you're allowed to enter. And that's where I'm saying, if there's a chance it might not be accepted, then you might want to save a draft of what you used to fill this in. And then you can think about what you could potentially revise in the future. So if the report is accepted, this is a screenshot of the MAUDE database. And I don't know, it might be a little bit confusing that you submit the report to FDA MedWatch and it gets accepted in the FDA MAUDE database, but that's just the way it is as far as I can tell. So on the next few slides, I will go through some specific examples where I submitted an FDA MedWatch report and then something was accepted and published in the FDA MAUDE database. So the first example is for Mayo GeneGuide. I believe this is offered through Helix, and this relates to a report that said I was not a cystic fibrosis carrier when I was a cystic fibrosis carrier. And if you read the details, you could figure out the explanation, but since they were focusing on a somewhat limited number of genes for disease risk, that I thought was worth emphasizing amongst these slides, and it was ultimately accepted to put it in the MAUDE database. So to explain this in somewhat more detail, at least from 23andMe, I could get a result that indicated that I was a cystic fibrosis carrier. So I don't have the disease, but if someone else was a carrier, then there's some chance that my children could have a disease. And so this particular Mayo GeneGuide result said I was not a carrier, but it had to do with the particular variant not being included in what they were reporting for the test. Now, the sequencing actually covered that. And if you looked at my variant calls, you could see that the variant call was correct and that I did have the variant that was the cystic fibrosis carrier variant. And with some helpful public discussion, I could learn more about the expert panels and the evidence to support the classification of this being a variant that causes cystic fibrosis. And by sharing, I believe, it was the CFTR expert panel results with the genetic counselor, that's where we came to the conclusion that I am a cystic fibrosis carrier. And so that's why I submitted this report where I received the false negative result. For all these slides, these were submitted a while ago, so I don't know everything that's going on with the companies now, but at least in this situation, I believe the product is discontinued. And also in the example above, you can see there are a couple of highlight examples where the formatting looks perhaps a bit strange, and that's basically where the FDA took out some things they thought could be identified of myself. I actually told the FDA they could tell, well, usually I believe I tell the FDA they can inform the company if they want of my identity, but that they aren't putting that in the public report. And so that's the way it looks. I think they also changed the capitalization, some things like the letter I, but I don't know why that is. But these B6s are the de-identification. So the next example is a bit more complicated to explain. So I'm going to describe Nebula low coverage whole genome sequencing. And so low coverage whole genome sequencing means you're getting some reads across your genome, but you might not have even one read at each position. And so they do this thing called imputation to guess what your genotypes are. And so this is one particular gene, APOE, and it's something that you might see somewhat frequently being described as something people would look forward to estimate their risk for Alzheimer's disease, or at least describe one of the possible risks. And so the APO gene, there's a model for the gene shown at the bottom. And with the exome data at top, you can see that there's this coverage. It's higher for the exons that code for the gene. And then at the bottom, there is a whole genome sequencing data where it's sort of mountains of coverage where it's not perfectly even coverage, but there should be some coverage across all the genome. The possible exception would be if the genome reference had a sequence that was not present on either copy of your chromosomes, then you wouldn't have reads. But usually you should have some reads everywhere. And so low coverage whole genome sequencing data could potentially have that if you sequenced it to enough depth. But what I'm showing you here is that there's this allele for the risk estimate for a risk assessment for APOE does not have any reads. So the result that the company provided, and I don't remember off the top of my head the exact details of how this was imputed, but essentially they could look for reads in the variants in other reads and say this nearby variant means you might expect the variant to be present at this particular location. And I believe there are two SNPs that are used to find these two different alleles, so that E3, I believe, is the normal allele, at least if you only consider, I believe, three possibilities that I can think of for E2, E3, E4. But I have one E3 allele and one E4 allele. And between these two alleles, the difference is the SNP at this location. So basically the reason it said I had E3 and I really have E3, E4 is because it did not guess the correct SNPs that should be present here for my genotype. And so this underlying issue can also affect polygenic risk scores. So that's what PRS stands for. And so the example on the left, I could describe with one sample, but I waited some extra time and I collected another sample. And because these are supposed to be the variants that you are born with, even if there's some time in between sample collection, you should be able to think these as replicates. And so you can see that there's a correlation with a correlation coefficient greater than 0.08, and most of them are more or less along the diagonal, but there are also some outliers. And so one of the things I emphasized in the report was that you can see here that one of my replicates said that for this particular score that my PRS percentile was 2, and the other replicate said 99%, and that's just about as different as you can get. So if that was important to you, there's some chance there could be a very different result if you just had another replicate, or in my opinion, if you just had higher coverage sequencing. That would hopefully resolve the issue, at least in terms of being more confident about the variant calls. And again, I haven't checked up in great detail for all these companies. My understanding is I think Nebula may be focusing more on the higher coverage sequencing now, which would make this exact point not as relevant, but I still think there are some ways you can get low coverage whole genome sequencing, and for humans and actually even non-human species, you can get these sort of ways to do imputation. So I think it is a concept that was worth keeping in mind. And if part of the goal of these slides is to get feedback from others and find others with similar interests as myself, I'm also trying to learn more about polygenic risk score, and both in terms of what can be a good, useful example, as well as what are some limitations. Although I think this is a little bit harder to describe for something like an FDA MedWatch report, but just relatively quickly try to go through a couple examples of the sort of thing I might be interested in. So I have a blog post where I describe a variety of different polygenic risk scores, and one of the first things to describe is a type 2 diabetes polygenic risk score from various companies, and I believe the polygenic risk scores themselves would put me at either average or above average risk. However, for one of the results, you were allowed to add in your BMI information. Then my risk dropped substantially, and I am almost 40 years old, and I don't have type 2 diabetes, so I think the estimate that may put me at lower risk when I consider my BMI is more accurate to my true risk as far as I can tell. And for the second example, I definitely know that there's more that I can do in terms of learning how best to use this bioconductor package to apply the iCare breast cancer PRS. But what I do know is that there's a minimum and maximum absolute risk that can be reported. And I think my sex of birth at being male is probably going to mean my risk of breast cancer is lower than even the lowest possible risk that became reported. And as another example, if your sex at birth had been female, I believe there are probably rare variants that could affect your risk even more than whatever is the difference in the largest or highest values that could be provided for the polygenic risk score. So in other words, there can be other considerations such as what is maybe not directly a genetic measurement of your BMI, or other genetic alterations that are not part of the polygenic risk score are things that you may need to take into consideration from a practical standpoint. But here, this is talking about issues with being able to get the right genotypes or the right variants. And if you can't do that, then I'm saying that's what called PRS issues, and that I'm pretty comfortable saying. So for Viome, that's a different company. You could do things besides metagenomics data research, which is in the sense that the metagenomics data is one example. That would be something like the bacteria classifications of bacteria that are in your gut. There was a kit where you could get both a stool sample to look at bacteria as well as a blood sample, but I did not want to collect enough blood for myself for that, so I'm only going to focus on the metagenomics part. Now, a factor in why I looked at Viome could be that I believe they had a FDA breakthrough device designation, although as I was having these discussions with the FDA to prepare for my submission, I learned that there's an FDA regulatory misconduct report. I don't want to say too much about that, but I will say that you have to be careful about whether you say something's approved by the FDA. So you might consider positive designation as being something that is considered favorable for the company, but the individual things that I purchased were not FDA approved. And if I understand correctly, I don't think even that designation is the same as approval. So that would be an example of a different form from the FDA if I'm generally trying to describe sort of things that people can participate in. But I'd really prefer to focus more on the FDA MedWatch results, which were more like technical issues in terms of what could be wrong with the results rather than how it's described and advertised. So to describe that part, which I think is the more important part that I'd like to focus on, well, not more important, but better fit for me to describe, is that on the right-hand side, you can see I've collected stool samples from different times. And in particular, I've highlighted these two columns where these are actually two scoops from the same sample collected at the same time. So if the idea is you're supposed to be able to take something about your sample and draw a conclusion about yourself as a whole person, then you'd want those to be consistent. And this example with the food to eat is probably not the most severe example, but at least it's a somewhat small table, and I can explain the idea, I think, relatively easily. And so one is that just the things in one column from the same sample are not identical to the things in the next column for the sample that was collected at the same time. And then I've changed the color of the font for some things. So for example, almonds are not exactly the same thing as almond milk, but I definitely drink almond milk on a regular basis. I don't plan to change that. In fact, for everything from any of the different types of results that I was given, I did not make any changes based upon what the report said. Maybe everybody doesn't need to be that strict. Some people have favorable reviews they've submitted, but that was what I did. I think there are at least some other examples where people might want to do something that's cautious like myself. And so another example is yogurt. I think that helps with my digestion, and I'm not going to stop taking yogurt because it told me to. To be fair, it told me that I should avoid drinking coffee, and this is something that I found to be important. I drink tea instead of coffee usually, and I think the coffee can irritate my eyes and maybe to some extent affect my digestive system. Although, again, in terms of limitations, I think that's kind of a general thing in that it's just most days I try not to drink coffee. And so I don't think there's anything occurring on this second sample or the second day of collection that was different the other days. That means I need to be even more vigilant about not drinking coffee. But the idea is valid in terms of me preferring tea over coffee. And so this is not the only type of recommendation that was given. And this doesn't have, I don't believe this was one of the results that had a score, but some of these results can have a quantifiable value that's associated with your result. That's supposed to, I believe, reflect for the signature across multiple bacteria or something. And so what I'm showing you is dots from scores from two different companies, and the scores can represent very different things, but there's a concept that I think was important for the FDA MedWatch report, which is that Thrive is a different company that also looks at metagenomics data, and they have two of their scores here shown in green, and the x-axis is the average value between the scores, and the y-axis is the standard deviation between them. So, and there's a very faint line that may be hard for you to see, but what I'm showing you is essentially that the Thrive scores were the highest average value, as well as having a standard deviation below this line that may be hard to see. And there are two other Thrive scores that are below that line as well, although if it's the same standard deviation, you might want to give higher priority to the scores that are higher value. It's not exactly the same as a standard deviation. Let's say a difference of 5 might be something to emphasize more if you're going between 5 and 10 versus say 90 and 95. But regardless of what you think about this point right here, these are the points that I was trying to describe for the FDA MedWatch report. So this means for the gray dots, it is these two samples that I collected scoops from the same sample, and I submitted them through different email accounts. And you can see that there's some noticeable standard deviation. The standard deviation is higher than these other somewhat more consistent results. And I don't remember what this means in terms of percentiles exactly, but if you could get an average versus a further from average result, and the difference could just be if you take another sample, either gets close to the average or becomes further from the average, then that is something that's, I think, important to take in consideration. And I wouldn't know off the top of my head which one of those was more accurate. There are some supplement recommendations which could potentially, at least in my experience, be something that you want to be even more careful about than the food to avoid recommendations. And I'll describe on the next slide an example of supplement recommendations from a different company, which is one of the reasons why, almost especially for the supplements, I did not actually try any of the supplement recommendations from this company. So for Vitagene, Vitagene's different than Viome in that they look at your genotype. So the variants that you have, or your parts of your genomic sequence that you inherited from birth. And so in this case, I did try a couple things. One of them was a zinc supplement, and I believe it gave me a relatively bad headache, but I didn't report to the FDA because I think the dosage for the tablet that I took for the generic tablet was higher, a good bit higher than was recommended. So I don't know if that was completely fair to describe. But what I thought was more fair to describe was that there's a recommendation to take theanine to improve everyday stress. And that's where I reported an adverse event for taking L-theanine. And to give an idea of what I mean by a moderate adverse event, I'd say if you have something like dry mouth for multiple hours or more, and if you have dry eyes, irritated eyes, for some noticeable amount of time, I consider those moderate adverse events, and that's within the range of what I was reporting for this L-theanine that I took based upon the Vitagene recommendation. And this report was accepted into the FDA, the FDA MedWatch report was accepted into the MAUDE database. And I think it's at least of some interest in that I don't think it said it on the Vitagene results, but I think on the product after I had the adverse event, I noticed that it says if you are currently taking prescription antidepressants such as MAOIs or SSRIs, consult your physician before taking this product. And I was taking and am taking SSRI. So SSRI stands for selective serotonin reuptake inhibitor. So I think the idea is that if you have more serotonin available to signaling, then that can help for certain things. And so if I understand correctly, I think it's possible that the downstream effects of increased L-theanine might relate to the effects of having more availability of serotonin for signaling. But you don't have to have this idea of a hypothesis of mechanism. I mean, I think it was something that was helpful to mention, but let's say it wasn't even a moderately bad adverse event. It was a really bad event. So this was not as bad, but to give you an example of a severe adverse event, which you can definitely report to FDA and definitely matches the reason of having an FDA MedWatch reporting system, would be a different drug that I took that it caused me to have I felt like a noticeably elevated heart rate. I didn't measure my heart rate at the time, although even with some amount of passage time before I went in for my follow-up appointment with my neurologist, they could take measurements and say something didn't look right, and they basically said, don't take that medication again. So that would be, even though it was an FDA-approved medication, if you experience an adverse event like that, you should definitely report it. And I don't know the explanation of why that was. But in general, when you fill out that detailed report, you say what other drugs you're taking, you provide other information. So hopefully that gives the information that FDA needs. I think they're allowed to contact you, although I don't think I've ever been contacted for more information. So after those slides, those may have been highly positive. So I want to focus on something I really like in terms of an option for genetic results. And part of the reason, there's two reasons I like it. One is that it's free whole genome sequencing, and one is I like the way that they're trying to set certain expectations with probabilistic language. So let me talk about the free part of it first, and then we'll talk about what this screenshot is showing. So for the free part, there's the All of Us program. You can select to participate, and then I believe, I don't know what's the chances, but I think there's a decent chance that you'll be selected after you ask to participate to have a biospecimen collected. And I believe currently that can be a blood draw or a saliva sample. I'm not sure. I had a blood draw. And then you can, I don't know if apply is exactly the right word, but I think of it like an application to get your whole genome sequencing from that sample that was collected. And my understanding is everybody does not get sequencing that got a sample collected, but after a long enough period of time passed, I did get sequencing for my sample. And so in itself, the idea that it's free sequencing, I think is something I think is really helpful. And even though I don't have access to my own data, which I hope can change in the future, researchers have access to do genomics research. And now let's get back to what relates to the previous slide in terms of this probabilistic language. Well, I guess if somebody had a range of estimates and could provide references to how those estimates were determined, that would be even better. But if you have a comparison between providing no clear warning and providing a clear warning, I definitely like the clear warning. So you can see here that it says genetic information is really just one piece of the puzzle. And so pharmacogenomics, I could consider something like the previous slide be pharmacogenomics, where it was recommending supplements to take. Some might only want to focus on FDA-approved drugs, but either way, it's your genomic information informing some sort of substance or drug that you're taking. And so it's saying that at least for what's being returned in this All of Us report, it won't tell you it won't tell us if the medicine will definitely work. It won't tell us if a medicine will definitely cause side effects or won't work at all, which I had to read carefully because this is actually a little bit different than the previous slide. What this is saying is something I'll bring up in a later slide to try to use your genetic information to try to guess. If you have to take some trial and error, maybe try to guess what not to take your first try. And as a matter of personal preference, I might actually be more interested in that. But the flip side is that if you make a positive recommendation of something to take, that can cause some sort of adverse event. I don't know what the probability of that happening is, but it definitely can happen. And that's essentially what that last example of Vitagene was. And then another thing it says is it won't tell us exactly how much medicine someone should take. And this is something I think has been important in terms of my personal experience. For example, I've seen a lot of different mental health professionals, and that can be difficult in terms of figuring out the right dosage and the medication for trial and error process. But at least in terms of what I'm thinking of at the moment, there is also something important in terms of what you need to take at a given point in time. In other words, there could be different environmental factors that can increase your amount of stress, and that can increase or decrease the amount of medication that you need to take. At least for me, if somebody has a more severe disease, maybe that's not as much of an issue. But if you're just trying to say what could happen in general, I would say that you couldn't use my genetic information at birth to know what medication I should take and what dosage that I should take for every year of my life, because that can change as different things happen to me, as well as I change the way I respond and process things that happen to me. And so I think that that's also something very important, and that is something that you can change. So I think that's a very important limitation to make clear. And also kind of consistent with, like, for example, I really said multiple times for Viome that I did not take any action based upon what it was saying. And so part of it was if I've already kind of gone through a trial and error process on something that works for me, it's not impossible that that could be further improved, but there's also a possibility that the recommendation was just not actually the best thing for me. And so this is saying, if your doctor's prescribed a medication for you, keep taking it and talk to your doctor before you change your medication. So there could be factors beyond what was taken into consideration for that report that are very important for you in your individual decision of what you should do. And so the next section I'll try to go through relatively quickly, because I don't know how people will be working with their raw data, either for sharing or for reanalysis, but I consider it something very important. So anyone else who has some ability to do some reanalysis or is interested in the raw data, I think it's something that's good to know about. So I think this PrecisionFDA system might have been built because of this challenges in the research setting. But what I think is really excellent is the infrastructure, which can be used for anybody. So, well, it can be used for anybody. Part of the reason I'm saying up top that I think of it as called controlled access data shares, you have to request access for an account. So even if you share your data with the most people you could possibly share it with, it's still only amongst the people who requested and been given access. But if you've met that criteria of having experience with genomic data analysis and being comfortable with working with the raw data, I don't think that's likely to be a problem. And so if you create an account, one thing is you can help other people find your data that also hopefully have experience working with the raw data. And you can also be more limited and share everything privately. Now, at the current time, I don't believe you're sharing data privately with the FDA in the sense that you're complementing something like the FDA MedWatch system, although that's something I think would be excellent. And I really hope this infrastructure could be used to do. But at the current time, what you could do if you really wanted to only share data, if you really wanted to only upload the data privately, you could reanalyze the data. So I believe you get about $100 to do that amount of data analysis for free. And I've reanalyzed at least two whole genome sequencing samples. And I haven't run out of funds to be able to do free analysis. So I think that's reasonable. And that is something you can currently do, even if I'm really pushing to hopefully have this as a way to help share raw data for large data, which is otherwise can be kind of hard to do. And so there are web-based and command line options. Again, command line options mean if you're not familiar with doing command line work, then doing things on the command line for your computer, then that might make this less of a good fit. At one time, both worked. Hopefully, they both work now for new people as well. But to kind of share my experiences and the troubleshooting for that, I did have some more recent experiences where the web interface could actually be better at sharing the variant call files or VCF files for list of variants that I believe you usually have for sequencing data. And to be honest, that's kind of what I think the PrecisionFDA has more of an appeal to be used for. And it's kind of weird because you'd expect the command line to be better for the larger size data. So if you have the same size thing, it's kind of weird for the web interface to work better. But that's at least my own experience. Hopefully that's not a common experience. And if you did experience what I experienced, then what I found is that the other people in the community were very helpful and important. So for the examples, these larger files like a BAM alignment file or FASTQ reads, I personally want to share everything publicly. So I have all this uploaded to the Google Cloud. And if you have a public link, which could become Google Cloud, or if you're quick enough, maybe you could even do from the company, which temporarily, maybe they share it with you publicly. If they don't share it with you publicly, then you've got to figure something else out. But they have a URL, I don't know if you call it a function or an app, but basically someone else developed this that helps solve the problem that I encountered. So if you upload to Google Cloud, it may not be free, and so that's a notable limitation if you're trying to have a completely free option to upload and share and reanalyze data up to a certain amount. But it does provide a way to help other people potentially find your data. And if for some reason you didn't want to keep it on Google Cloud, it could still be there. I don't know which is more likely to be here in 10 years, but I hope both are. And so I also hope that even though I make my data public, this increases the chance that people can find my data and find something useful to do with my data. Okay, so last section is a bit complicated. I'm really talking about three things. So one is NIH ClinGen, one is Genome Connect, which is something that's offered through ClinGen as a way that you can upload your PDF files, and there can be some functionality that I'll describe later. And I'm going to describe why I was even looking for something like NIH ClinGen Genome Connect, because that's what I'm really interested in learning more about at the current time. So to start with ClinGen, you kind of see this interface where you can look up information about, for example, a particular gene, and I'll give a couple examples about that. But getting back to the kind of overall theme, there's a link for data sharing resources, but as I say here, it's actually for clinicians and laboratories. So if you're an individual and you want to find a way to participate in data sharing, you'd want to go to Genome Connect. And so BRCA1 is a gene that some people may be familiar with, and that it can have variants that can increase your risk of breast cancer. And in general, this hopefully gives you a sense of what ClinGen is supposed to do. So you've got different types of information that can be provided for this gene. And the one I'm going to focus on is this variant pathogenicity. And so I've gotten some very helpful feedback from the Genome Connect staff. And my understanding is that when there is an expert panel evaluation for a variant, that creates 3-star evidence for that variant in ClinVar. So ClinVar is the database I was more familiar with before I heard about ClinGen, but this means you can get this sort of information from both of them. And then one thing that I found, I think is very important to say, is that even if a lot of people are familiar with the disease risk that can be associated with some variants in this gene, if you use ClinGen, there's actually a lot of work that needs to be done by Enigma for annotating the variants. Right now, there's only 20 classifications for pathogenic, likely pathogenic, or uncertain significance, or benign, but there's a lot more than 20 variants that have been observed for BRCA1. So at the current time, my understanding, and again, this is as somebody who'd like to learn more, but at least my level of understanding, that there's a resource called BRCA Exchange, and ClinVar also has the variant classifications from Enigma, just that they're, if I understand correctly, they're from an earlier point in time, and now I think Enigma is trying to participate through ClinGen. So that's probably kind of complicated if you've never heard about any of these sort of things. So I'm going to go through 2 variants to try to explain it, hopefully in a little bit more detail, or at least make it a little bit easier to understand with specific examples. So these are not exactly the same type of links. So you can see ClinGen, the logos on both of these, but if you look at the web link, then it's actually a little bit different for these. And I'll get into that a little bit, but I mean, if you look at them, these 2 pages, they look similar perhaps at the top. If you scroll down, this is one of those 20 variants where there's a pathogenic annotation, and I need to learn how to understand this variation a bit better, but there's some information about the evidence for that classification. And then this is a different variant. If you scroll down, you don't see that same pathogenic label, but it is in ClinGen. And so I found this using BRCA Exchange, and then you can click right here to see the variant in ClinVar, and you can see it has a pathogenic classification. And if you scroll down, you can see that there is 3-star Enigma classification information. There's a 3-star classification from Enigma. So Enigma is still providing a level of a certain level of confidence in a classification of this variant is pathogenic, just that Enigma has not, as far as I understand, Enigma has not gone through that same process through ClinGen for this variant. Okay, so let's go to the next slide. So CFTR is another gene. CFTR is a gene that's associated with, it has variants that are associated with, not associated with, can cause cystic fibrosis in a recessive manner. And again, this kind of shows you that there's multiple types of information that are communicated for a gene search in ClinGen. But one thing I hope you'll notice is that if you go to the previous slide, this is the variant pathogenicity for BRCA1. You don't have a section like that for cystic fibrosis or CFTR for cystic fibrosis. So that's not because of a lack of confident gene annotations. I think it just means that there has not been a ClinGen expert panel submission. Now, if you look for variants within ClinVar, you can find expert panel evaluations, and that goes back to the earlier discussion of submitting the FDA MedWatch report for my cystic fibrosis carrier status that was reported as not being a carrier in one particular result. So hopefully this will change over time. But even within NIH resources, there are other ways you can find more information about a particular gene of interest. And hopefully this will be including information about CFTR for the pathogenicity for cystic fibrosis before too much longer, I hope. Okay, so now we're going to move back towards trying to get to the topic of Genome Connect. And unfortunately, I'm going to take another slightly tangent conversation and then get back yet again. But this is one of the next screenshots you would see in terms of how you would actually access Genome Connect. And now I'm going to talk a little about, well, why I was even looking for Genome Connect in the first place. So I have participated in something called the Personal Genome Project. So you can see completely public raw data as well as reports uploaded, but I encountered some issues with that more recently. And I also have a GitHub repository, which I'd say people commonly use for sharing code for reanalysis, but you can also share other similarly sized files. So now you'll see a bunch of PDF reports or screenshots on the GitHub repository. But the GitHub repository, even though someone could in theory take GitHub repository links from a bunch of individuals, and that's how you could kind of help find that data that was shared publicly, it's not a very systematic way. Different people might have different ways they'd want to share their files. So I was hoping the NIH was doing something that kind of worked like the Personal Genome Project or PrecisionFDA or others to kind of have a way to systematically share data in a way that you can understand what's being shared. And I believe Genome Connect provides this goal, although I believe it provides something different than what I'm currently doing. So I'm still going to provide information on GitHub. I'm going to show you, hopefully somewhat briefly, an example of that so you can understand what I still am doing and what I was trying to find an alternative for. And then I'll finally get back to the topic of Genome Connect. So to explain what I have now and what I think can be of use to people who want to have examples of results before they try something out, you can see this is a screenshot from my GitHub page, and this is not how other people necessarily going to do it, but this is how I do it. Is I have folders for different companies or organizations. So for example, you can see All of Us, which is that NIH option for free whole genome sequencing if you're selected, and Genes for Good, which is an option for free SNP chip genotypes from the University of Michigan, as well as other companies and organizations that you may have heard of, such as 23andMe and Ancestry DNA and so on. So this can be interactive, and you can click a folder and see different files. You'll see some scripts like Perl or script or R scripts, as well as PDF files or screenshots. So that's what I'm doing right now. It's completely free. well, GitHub is free, and it's freely available for other people to see. And I'm not going to go through too much more examples of how to use my GitHub resource, but let me show you what I did for that NIH All of Us Pharmocogenomics example. So there's an earlier slide where I took a screenshot and I showed you that screenshot, and you should see part of what I have there here. So I have it in italics and in quotes because these were the exact same sentences from that screenshot before. And I changed things around a little bit. So it's a bit of a stretch to say it's very similar to what Genome Connect staff are going to do, but it has some similarity in that there are, I think these are PDFs I created by essentially trying to have a website that I printed as a PDF instead of printing a sheet of paper on a printer. But I could upload a PDF, and then I essentially kind of parsed out information from that. So before I described how I had an adverse event from a drug that was recommended, but the warning that they won't tell you it will definitely cause side effects for retinol all is actually, I think, more in mind with what we see here. So in most cases, it didn't have anything that it said, but for this particular gene, it said that the gene was expected to have decreased function, which may increase your risk of developing muscle pain. So I've never taken this particular drug before, but this was that probabilistic language, may. So it doesn't know for certain it's going to cause a side effect, but there's some evidence that it may increase the likelihood that going to have an adverse event. And so I just thought this was kind of a different way to show you the results, and the idea that someone could parse something and show it to you in a different way is helpful. Now, at the current time, I don't think it's exactly showing you it, but I believe behind the scenes, you're getting functionality that you did not have from the PDF or your web-based result that you originally had. So that's where there's some similarity, although everything I showed you for GitHub is completely public. Everyone can see. That's not what Genome Connect is doing. But for some people, in fact, most people who don't have as much experience with genomic data analysis, I would probably recommend something that's more controlled access that's clearly public. But for me, I hope sharing it is helpful for you and others. Well, it's helpful for others, even if you have experience with the analysis, or if you don't have as much experience, if you at least understand the sense that there can be these PDFs or screenshots that you can see what my results look like. Okay, so now we're getting back on topic. So this is the Genome Connect webpage, and this is where you have to get if you want to participate in this program. So let me go through the steps of getting to this webpage, because if you started the way I started, I think it's helpful to show you how to get there. So let's say you start from ClinGen. This is the NIH resource, and that's, I believe, where I started. And so there's a link for data sharing resources. Again, we're not going to focus on the way to search for information for this particular presentation. So we're going to go to data sharing. And so this is where I said information for laboratories and clinicians, which I think is very important. I'm very glad they do. But for individuals like me, you want to go to Genome Connect, and then you say for individual patients. And then you click on this link to Genome Connect homepage. And so now this is where you need to be, where you can sign up to register for an account. And after you do that, you can log in to participate. So this is something I am very interested to learn more about. So one thing that I couldn't do it for, actually, I couldn't complete the registration at first. So I was very helpful in terms of I got prompt responses to explain that I can register myself under other disease or health conditions. I looked for these kind of general diseases or conditions like anxiety or migraine, and I couldn't find any match. And I'm not 100% certain, but I might guess that if there are advantages to being able to find individuals for sufficiently rare diseases, then that's why it might help to have a very specific description, because you can search based upon diagnosis. And if you search by migraine or anxiety, that might get you a lot of results. But if you search for a diagnosis for this rare disease, then that might be getting you to a small number of people relatively quickly. At least that's what I think could help. And in terms of the gene search, I'm still trying to understand exactly how this is working to kind of go, if you thought about what I did for that NIH report, where I looked at a report, I created a different format way of representing what's in the report. And then that's the sort of thing where I don't know exactly what's going on, but I can understand the benefits in the sense that, let's say you have a rare disease and you want to find other people who have variants that could be the same variant, or at least another variant in the same gene. Assuming that other parties worked out of how someone parsed your files and made it searchable, I can see why this could be of value to help you connect with those other people, where it may, I'm not sure if you, how easy, whether there's been a submission to ClinVar, for example. Maybe there already is, but I don't know all the details for some of these more rare diseases. And for geographic locations. If it was not a rare disease and there's a lot of people who have variants in the gene, then maybe you want to emphasize, maybe you'd like to focus more on the people who are closer to you. Now, I was talking about how this is preferable for being more controlled access than sharing everything publicly. And if that is true, then you could also have some people who don't really want to find the other individuals. So that's where I might ask the staff to help make sure that you're participating the way you want to participate. But even if you didn't want to find other individual people, there are ways in which participation can help. So I don't have experience with this in my own files, but my understanding is that Genome Connect is part of ClinGen because they're helping you connect not only with other people, but with the publicly available information in ClinGen so that you can receive free variant updates. What that means is, let's say you have different results of different companies, or you have a company's result where the company actually changed what their classification was for a variant over time, but you got your result before that change occurred. Genome Connect staff are at least capable of parsing your files that you upload, and if there's a difference and that difference is reflected in ClinGen, then they can send you a notification and you're going to be informed about this for free. Now, this is really appealing and really interesting to me, but the staff did want me to make clear some limitations. One is that it's not known what's the timeline, that if they do provide you this update, how long would you have to wait between the time you upload your PDF or whatever file is acceptable to be able to get that update. Also, they wanted to make clear that most participants will not receive updates, and Genome Connect does not identify all genetic updates. So for the second part of that, I'm not sure if I exactly understand what that means for specific variants, but I could imagine that, let's say, for example, you wanted to focus on rare diseases and maybe not as much on the pharmacogenomics results, or if you did do updates for some pharmacogenomics results, you only focus on the ones that weren't predictive. I could see why maybe with a certain amount of staff working on the task, maybe you prioritize some tasks over others. And for the most participants who do not receive updates, I guess that relates in part to how often you get these changes, and those changes can be captured in Genome Connect. But that's where I could imagine this being something that could be quite helpful. So if the timeline was short, which it may not currently be, but you had, say, contradictory results and you want to try to get an understanding, if the staff at Genome Connect can help you understand what's in ClinGen better by basically saying, I think this is the result that there's better support for, then that's helpful. I guess I think it's helpful to have from the NIH staff, but what's very good is that even if you don't have a quick update, a quick timeline to be able to do this now, at least it would be all based upon the publicly available data in ClinGen. So if you were familiar enough with ClinGen, or if you made an appointment with a genetic counselor who was familiar enough with being able to parse some of the evidence for the variant status to try to understand that contradictory result, then that could be helpful. And hopefully, if there could be some way that this could work that complements whatever the genetic counselor be doing with you, is what I could imagine could be very exciting. Another thing would be, it's exciting is not exactly the right word, but it still would be important, is let's say the classification changes over time. So my understanding is this doesn't happen very often because it says most participants do not receive updates, and there are other reasons why I don't think it should happen very much. But let's say, unfortunately, the classification changed so that you were told that you had a pathogenic variant, but it was changed to be benign later on. I mean, I don't think I think the most I've seen for myself reported, no, the most I've seen reported in a publication, I think it's been between likely pathogenic or likely benign. But I don't know if you actually could go all the way pathogenic to benign if you saw enough variants or you waited enough time. But to explain why it could be important, even if it doesn't change your own care, I mean, someone later on, if you had children that could inherit the variant, if it was actually not a pathogenic variant, you'd want them not to do the same sort of things that you do if it was a pathogenic variant. And that might even mean you might want them to try out newer technologies to see if something else can be found that is associated with their disease risk. Again, I don't think it happens very often, but if it happens at all, I think this could be an extremely important resource. And this is something where I think it's better to talk with a genetic counselor, but if you have talked to a genetic counselor and you're talking to the genetic counselor because you have a family history, then I would imagine that you can even further reduce your chance of this happening is if you're in your own family, the people who had the disease have the variant of interest, and you basically kind of validate it within your own family. I think that's something that is helpful, and I think that's part of the reason you're supposed to talk to a genetic counselor to figure out how you can maximize your confidence in what decision you want to make. And that would be, I think, one example is you look at other family members' genetic results. And so if you're familiar with publications, there's a publication that describes ClinGen and Genome Connect. And within that publication, they say that Gene Connect is meant to help the goal of ClinGen to assist with supporting data sharing. And this is exactly what I was looking for, something from the NIH to support data sharing. Now, I was doing it in a completely public way, and the way they're doing it is different, but it's also good to learn about the other possible ways that you can participate in data sharing. And it also describes some more about details of what it can do to help the community in terms of how, even though they're not providing that information about you to give you personal information put in the public, they are keeping track of who has the disease and which variants are present in the person with disease, and they at least have the ability to add variant annotation, variant classifications in ClinVar, and or refine the variant information for the classifications in ClinVar. So that is used to the greater community, as well as the functions I described on the previous page of helping you learn more information to make decisions for yourself or your family members. So I think this is great. I hope this can continue and be expanded. And I hope you can also get similarly helpful information as I received by sending an email to either clinicalgenome.org or info@genomconnect.org. Okay, so thank you very much for your interest. In general, I want to try to find other people with similar interests as myself. If you have additional feedback you think would be good to take consideration, I'd very much like you to add some sort of a comment. And I hope I help share some information, at least some people, that they can learn how to make better use of their data or share their data to help other people. All right, thank you again.